So this next video is on RNA structure, so the structure of that single copy of a gene that codes for a protein. The RNA nucleotide looks a lot like the DNA nucleotide, but there are a few very important differences. So the RNA nucleotide also has a phosphate. Here's one of the differences. It has a ribose sugar, instead of a deoxyribose sugar. So RNA is ribonucleic acid, DNA is deoxyribonucleic acid, so ribose sugar in RNA. And then the third component is nitrogenous base. Just like in DNA, it has the same exact nitrogenous base on its three-part nucleotide. So in general, a nucleotide is a sugar, a phosphate, and a base. The difference is, depending on the nucleotide for what, the sugar changes. So think about ATP too. ATP is a nucleic acid. It's made up of nucleotides. The difference here is that ATP has 3 phosphates, a ribose sugar, and a base to the side. And then RNA has a phosphate, a ribose sugar, and a nitrogenous base to the right. DNA has 1 phosphate, a deoxyribose sugar, and a base to the right. So the nucleotide structure is always a phosphate, a sugar, and a base. Okay. One big difference in RNA is its structure and the types of nitrogen bases that RNA has. So the first one is the same, adenine. Except adenine has a different partner in RNA than it does in DNA. Adenine, instead of pairing with thymine, is going to pair with a molecule called uracil. So uracil is very similar to thymine, but it's a different chemical component, so it's different entirely. So A with U in RNA, A with T in DNA, A with U in RNA. The other two are exactly the same. Guanine always pairs with cytosine. So the only difference in the nitrogen bases of RNA is that A pairs with U. The RNA molecule does have some differences from the DNA. The first is that RNA is only single-stranded. So remember we said that DNA can never leave the nucleus because it's too big. Well, part of the reason that RNA is single-stranded is so it can leave the nucleus. RNA is made by copying a segment of a single strand of DNA, so it is not the entire genetic code, it's just a piece of it. It uses uracil instead of thymine to bond with adenine, and it leaves the nucleus to go to the ribosomes in the cytoplasm. So that should tell you if it's going to the ribosomes, it must be used in making proteins. So the RNA molecule can leave the nucleus, and its single-stranded nature is what allows it to do that. Now, there are a few different types of RNA that become very important when we're talking about making proteins. So the very first type of RNA is called messenger RNA. It is a copy of a gene sequence from a single strand of a DNA molecule. It's just a copy of one segment of RNA. When it is done, it's the part that leaves the nucleus and goes to the ribosome, either in the cytoplasm or on the rough endoplasmic reticulum. The second type of RNA is ribosomal RNA. This is a molecule that makes up the ribosome, makes up that protein building unit. It helps to make proteins during a process called translation that we'll get into a little bit later. The third type of RNA is transfer RNA. So all of these we refer to as an abbreviated, so messenger is mRNA, ribosomal is rRNA, and transfer RNA or tRNA helps to transfer amino acids to the ribosomes when they're needed. So mRNA is a messenger. It takes the message to the rRNA. rRNA is the ribosome, And tRNA is the transfer. It transfers amino acids to the ribosomes when they need it. So this graphic kind of shows you how all three work together. So at the top you have the rRNA, which is the ribosome. In the middle you have the messenger RNA, and that's the blue strand on the bottom in that picture. And the messenger RNA is what the tRNA is using and reading to bring amino acids to the ribosome. And remember, amino acids are the building blocks of proteins.